Hello, welcome to the Excited Education channel, the YouTube channel which brings you about mathematics and biology. As usual, I'm teacher Chris on my channel. Now we are going to discuss about, or we are going to continue where we have stopped at last session on the point what we call viral replication, and we are going to see what you call bacteriophage growth curve. I would like to invite each and everybody who is watching this video first to make subscribe, comment, share and like on my video to follow next videos. So let us advance here we have stopped to where we have stopped at last session. So what you call bacteriophage growth curve. Bacteriophage growth curve, the rises of we are going to first to know the periods or the stages that are required to make the bacterial growth curve, bacteriophage growth curve. So the rises of bacterial cell released a large number of phage simultaneous erratic replication cycle exhibit a one-step growth curve. So here there is the bacterial cell likely to release a large number of phages simultaneously, and the erratic replication cycle exhibit as one-step growth curve. So the eclipse period is the time between, yes, it is in the, these are the periods or the stages or the parts that are involved in the growth curve. There is what you call the eclipse period. It is the time between entry of the phage nucleic acid into host cell and the formation of the complete assembled phage within that cell. So if you take the first stage where that phage likely to bind on the host cell, starting from the point where it likely to release those nucleic acid from the phage into the host until the stage where the stage is already completed is what you call eclipse period. So the latest for the latent period is the time between entry of viral nucleic acid and the release of viral progen. So if the progen is already released out, since the viral nucleic acid is injected into the host cell, is what we call progeny process. Into the stage of what we call viral progen is those period between is what we call latent period. The burst size is the average number of phage released with the rises of one host cell. You know that if the bacteriophage replication complete, it can't leave the host present or the host as a living organism. Already the host is have to burst in order to release those phages, and the burst size is what we call average number, the number of phage released with the rises of one host. So here let us try to emphasize this curve by grouping those periods in one curve, one step growth curve. So red line is the number of the virus, what you call the unadsorbed viron plus those released from the host. This is what you call red curve. This one is it's representing the number of free virus, and the blue line is the number of total virons. This is what a number of total virons that are made. Here we have time, minutes and phage count, and latent period is composed of eclipse and intracellular. So here latent period, this is what you call latent period. This from here and here, you know, this and here. So this is what you call eclipse. So this eclipse is composed is composed in the latent period, and the latent period is composed of eclipse and intracellular accumulation phases. So here we have seen this on the figure as the life cycle of phage T4. How much time is the eclipse period and the latent period of the phage T4? You have to understand this. If you understand the process of eclipse, you find that it is composed of latent, it is the blue line. We say that is represented by blue line, and those blue line is comprised of total virions, and the latent period is composed of the eclipse and intracellular accumulation phases. So here, bacterial growth curve, when E. coli, what you call E. coli is what you call Escherichia coli, is infected with T2 phage at 37 degrees Celsius. The growth plateau is reached in about 30 minutes, and the burst size is approximately 100 or more virions. So this is in T2, but is infected with with E. coli, Escherichia coli. They likely to burst or to reach in the burst size about 30 minutes left, and they release more than 100 virions. The eclipse period is 11 to 12 minutes. You can ask yourself how many or how long is the eclipse period occurred. It's to take 1 to 12 minutes, and the latent period is around 22 minutes in length. And here, colonized microphage of Escherichia coli with assembled bacteriophage inside the host. This is the structure of colonized macrophage of E. coli. This is what we call E. coli with assembled bacteriophage inside the host. These are assembled bacteriophage inside the host. And temperate phage, Risogen. Some DNA bacteriophage are called temperate. This is what we call DNA bacteriophage. They are likely to be called temperate because they can infect host cell without replicating and enter phage and without causing rises of the bacterial host. This is what we call temperate. That's why we call it temperate. Those DNA they are likely to replicate, or they are likely to infect they call temperate because they can infect host cell without replication, and they can move out of the host without bursting of that host. Then rather, the temperate phage often establishes a state of Risogen. So this occurs when a temperate phage DNA is incorporated into bacteria DNA. You have to know that the viral DNA incorporated is referred to as prophage, Risogen, and rhizogenic bacteria are bacteria that have prophage on their DNA molecules. And you know that in that prophage is replicated with the bacterial DNA during the formal host. Cell DNA could replicate, and the host cell survive and reproduce. So here in the Risogen process, what we call is a genic conversation. This is what we call phage with bacterial chromosome. They inject those viral DNA. After injecting the viral DNA, they try to move or to develop, and they release what we call protophage. The rhizosis conversion occurred when the temperate phage DNA is incorporated into bacterial DNA, and the prophage viral DNA incorporated, and then rhizogenic bacteria, the bacteria that have prophage on their DNA molecules, they likely to have those rhizogen, and the rhizogen, this is state that viral DNA incorporated into the bacterial chromosome. This is what we call rhizogen. This is the state that viral DNA incorporate into the bacterial chromosome. This is what we call viral rhizogen. So temperate phage rhizogen. This is pointed directly to look some characteristics of rhizogen. What comprises rhizogen, or if you see rhizogen, what characterizes rhizogen? Stability. According to the stability, prevent infection of host cell with the same phage. The host cell is protected against the rhizosis ability of spontaneous rhizosis, frequent 10 to power negative 2 to 10 power negative 5. So here, in the rhizogen, it's have ability of inducible rhizosis, and its immunity. It characterized immunity, and it has ability of resuming some new physiological properties other than unaffected cell, and other than unaffected cell when some prophage genes are expressed. For example, bacterium like Corynebacterium and Diphtheria So, and you have to know that when they are in state of Risogen, because one of the prophage genes that is expressed codes for this protein toxin, it is the production of diphtheria toxin during an infection with Corynebacterium diphtheria. So here, what you call some properties conferred by prophage. So microorganism, medical importance and properties coded by phage. This is what we call microorganism, medical importance and property coded by phage. These are likely to read this, zoom it and likely to watch it there. These are the microorganisms. Then these are medical importance, what they likely to use in medicine. And here these are called property coded phages, synthesis of the diphtheria toxin and so on. So here let us continue on prophage. Can be passed from one generation of host bacteria to the next indefinitely. But once it is excised from so in this case the phage can establish as a erratic replication cycle and with reproduction of phage progeny and rises of the host. And this is the process of replication of the phage, and this occurred in different ways, where we starting and the phage injecting their genomes inside and then end up rising the phage is out, where it out comes to the bursting of the host cell. And here is what we call how it takes place to be or to this to reach on the point of the host cell or dying of host cell. If you have viral infection, then into the host cell, then host cell, disease of host cell and genetic alternation of host cell. Here in the part of disease of host cell, there is what we call productivity infection, more viral produced, then latent stage nucleic acid of virus become partial of the host cell. In the productive infection, more virus produced. Here they are what we call lysis of cell, release of virion and lysis of virion and lysis of cell. So this if the cell rises, the host die, and if here the host cell multiples, continue linkage of virion. Here there is what we call release of virion, but there is in non-release of cell. So the latent state it ends up when host cell is often modified and multiplies. And here there's what we call replication of plant virus. So allow me to stop from here, then we'll continue at the next session by explaining about the replication of plant virus. Thank you for watching me.